So, now that we have a solid handle on the two-sample t-test and the paired t-test, we are now going to move on to something a little more interesting, the comparison of multiple samples. And we do this by performing what's known as an analysis of variance. So an analysis of variance can test whether or not multiple groups are different from each other. A one-way ANOVA, which is what we're going to be going over here, is a test of whether the means of the groups are equal by assessing whether individual samples from different groups are more different than individuals from within groups. The hypothesis for a one-way ANOVA is very simple. The null hypothesis is that the mean of group 1 equals the mean of group 2, and that equals the mean of group 3. The alternative hypothesis is that at least one mu, at least one population mean, is different from the others. It doesn't tell us which one, it doesn't tell us how many, but it tells us that at least one is different. Then ANOVA is used to test if there is variation between groups beyond that expected by just random chance. We need to utilize some specialized measures for variation to make the ANOVA work. The first is called the group mean square, or MS group, and this is the variation between groups. We also have the error mean square, or the MS sub error. And this is a variation within groups. And this is similar to the pooled sample variance in two-sample t-tests. You can almost think of ANOVA as a multiple two-sample t-test. We compare the mean squares using the F ratio, which you may remember from the F test. We're checking the differences in the variance between two samples. And so F is the mean squared of the group divided by the mean square of the error. If F is close to 1, that will indicate that there's little difference between the groups. If real differences do exist, they should result in F larger than 1. So let's take a look at the calculation of ANOVA, because it can get quite complicated. The first thing we need to do is partition the sum of squares. And we can think of individual observations from each group as y sub ij. It's an observation of individual I in group J. The total sum of squares is the combination of the sum of squares of the groups and the sum of squares of the error. The sum of squares of the group is the sum of the sample size of the group times the average of the group minus the total average for all groups, also called the grand mean squared. The sum of squares error is the sum of the variance of each sample times the number of observations per sample minus 1. The sum of squares error is the sum of the variance of each group times the number of observations per group minus 1. And this is also the degrees of freedom per group. We get the total sum of squares by adding the error sum of squares and the group sum of squares. The first thing we need to do to figure out the group sum of squares would be to calculate the grand mean. And the grand mean is really easy to calculate. It is simply the sum of the sample size of each group times the mean of each group divided by big N, which is the total number of observations in all groups. So this can be a little bit daunting, especially in this format. And I want you to note that in this format, R cannot read it. So instead, we need to do it all out individually, all the sums out individually. And so let's take a look at our iris data and calculate our sum of squares based on some comparison we want to make. So let's say we want to compare the sepal widths of all 3 iris species. We first need to separate all 3 groups from each other. And so we know there are 3 species, Setosa, Versicolor, and Virginica. And so we make vectors of observations of sepal width from each of those species. We then figure out their n, their mean, and their variance. All of this stuff should look familiar. You've done this tons of times. And we do that for every single species. Notice that I have specifically given each one unique names. So N1 is specific to Setosa, N2 is specific to Versicolor, and N3 is specific to Virginica. And this is consistent through all of these calculations. We then need to take our grand mean, which remember is the sum of the sample sizes of each group times the average of each group divided by the total number of observations in all groups. And so first, let's just figure out big N. So that's simply the sum of N1, N2, and N3. That makes sense, right? And so y bar total, or the grand mean, is N1 times y bar 1 plus N2 times y bar 2 plus N3 times y bar 3 divided by N. And this gives us 3.057333, and that's our grand mean. We can now use that to figure out the sum of squares of the groups. Sum of squares groups is N1 times y bar 1 minus the y bar total, or the grand mean, squared, plus N2 times y bar 2 minus the grand mean, squared, plus N3 times y bar 3 minus y bar total squared. And this gives you 11.34493. The sum of squares error is very simple. All you do is take the variance of each group, so in this case, variance of the first group, times the degrees of freedom of that first group, plus the variance of the second group, times degrees of freedom of the second group, plus the variance of the third group times the degrees of freedom of the third group. And you should get 16.962. Then we can get sum of squares total, which is simply our sum of squares error plus our sum of squares group, and that is 28.30693. Now, since we have the sum of squares for both the error and the groups, we now need to calculate our mean squares, because that is what we're actually going to use to run the F-test, to run the Anova. The mean squares of the group is simply the sum of squares of the groups divided by the degrees of freedom of the groups. And we can calculate the degrees of freedom of the groups really easily by taking k, which is the number of groups, in this case 3, minus 1. And so degrees of freedom of the groups is 2. If we calculate out the mean squares of the groups, we get 5.672467. The mean squares of the error are the sum of squares error divided by degrees of freedom error. And the degrees of freedom error is big N, which is the total number of observations, minus k, which is the number of groups. So in this case, our big N is 150. There are 50 observations for each group, and then we subtract 3 from that, and our degrees of freedom of the error is 147. If we calculate the mean squares of the error, we get 0.115. We can then run our F-test. And the F-test is the ratio of our mean squares of our groups divided by the mean square of our error. And if we run that, we get 49.16004. If you notice, this is a whole lot larger than 1. So, likely, our groups are different from each other. We can test this by calculating a p-value using the pf function, and we include f as our q, as our variance ratio. Our degrees of freedom We have 2 degrees of freedom. We have the degrees of freedom for the numerator and degrees of freedom for the denominator. The degrees of freedom for the numerator is k minus 1, and the degrees of freedom for the denominator is big N minus k. You can double check this by looking at how we calculated the mean squares for groups or for error. And then, since this is an F test, we are only interested in the upper tail, so lower dot tail equals false. Now we get a result of 4.49 times 10 to the negative 17. This is extremely small, and so this means that, of course, it's smaller than 0.05. We reject the null that the means are the same. At least one of these means is different from the other. At least one of these groups is different from the other. Now, of course, all of that work is condensed in the Anova function built into R. And the function is AOV for Anova. And the way that we write out our Anova function is we give it our y value, the measurement we're interested in, and then we put in this tilde symbol, and then we give it our x value, the grouping variable, the information that it needs to know which group is which. In this case, our y variable is sepal width, and our x variable is species, and we know there are 3 species. We then also tell it where to get that information, where to get the data, and that is from Iris. And so if we run that, we get some interesting information here. We get our sum of squares. Oh, look, 11.34493. For our species, this is our sum of squares for our group. If we go up, we can see, indeed, that is our sum of squares for the group. Our calculation was correct. And then it also gives us a sum of squares of the residuals. Here, residuals refers to the error. And so the sum of squares residuals is also the sum of squares of the error. 16.96200. And there it is. So we calculated that correctly as well. We also have the degrees of freedom, 2 and 147. That also matches our calculations. And it gives us some residual standard error that we can ignore. Now, the one thing about this output, it's not very descriptive. It doesn't give us a p-value. It doesn't give us our mean squares. So we need to produce what's called an Anova table. And an Anova table in R is produced by using the summary function, which we used previously to look at summary statistics for individual vectors. And so if we simply put the name of our Anova, so up here we named our Anova model.1, just put that name in the summary function, and it produces a very nice Anova table, which includes information on our group, which is species, and our error, which is residuals. You can see the degrees of freedom are correct, 2 and 147. Our sum of squares are correct. Our mean squares are here, and that's correct. Our F value is also correct, and it gives us a p-value. Notice the p-value here is different. The only reason why it's different is that in these built-in functions, R will not give you a p-value that is less than 2 to the negative 16. For R, that basically means it's really, really, really tiny. You would report this as p is less than 0.001. And the results are consistent with what we found from our hand calculation. There is indeed a difference between our groups, though we're not sure which ones are different. Now, we can go a little bit further with this information because there's a lot of information in this Anova. And one of the interesting things that we can pull from an Anova is an R squared. And R squared is basically the variation that can be between groups that can be explained by differences between the groups. And it's a very simple calculation. R squared is the sum of squares of the groups divided by the sum of squares total. So it's just a simple proportion. We get an answer of 0.4007828. What this means is that around 40% of the variation between groups, 40% of the variation that we observe is explained by group differences alone. So that also means that there's 60% of the variance that we don't have an explanation for. There are 3 important assumptions for the one-way ANOVA. They are the measurements in every group represent a random sample from their population. The variables are normally distributed in each of the populations. This is a parametric test. And the variance is the same in all populations. This should look very familiar because it's the same assumptions as the 2-sample t-test. What's nice is that it is really easy to test the assumptions for ANOVA in R. After you run your test, you can plot your model, and it will give you some diagnostic plots. So here, first, we want to run par mfrow concatenate 2, 2. This will give us 4 cells. And then we plot our model. So we plot model.1. This is the same format as the summary function. And we run that. And you can see we have 4 different plots here. We have a residuals versus fitted, normal QQ, scale location, and constant leverage. The residuals, fitted, scale location, and constant leverage, these plots, the only thing you need to know about them is that if you see any sort of distinguishable shapes like squares or diamonds or triangles, then your variance might be different between your groups. If we take a look here, though, there's really not a whole lot of shape that we can see, any distinguishable shape we can see, so it's probably fine. More importantly, also, we can check for the normality by looking at this normal QQ plot. A normal QQ plot is basically a regression, which we'll talk about in a couple weeks, of the residuals, or the error. And for an ANOVA, you want your error to be normally distributed. So if we take a look at all of these points, they make a very clear straight line. They're not S-shaped, they're not J-shaped, they are shaped like a line. This basically tells us that, indeed, our error, and therefore our data, is normally distributed. So, using ANOVA is appropriate. And it happens that ANOVA is very robust to violations in the normality assumption. So, in most cases, ANOVA is appropriate. There are alternatives to ANOVAs, and we'll talk about that a bit in a couple weeks. One thing I want you to remember is that once you've run your ANOVA, the only thing you know is that the mean of at least one of your populations is different than the others. You don't know if there are 2 that are different, 3 that are different. You have no idea which ones are different. And so you actually need to go further and do what's called a post-hoc test. And post-hoc tests basically compare each group individually to each other. You could do multiple 2-sample t-tests for this, but it's really easy to do what's called a 2-key Cramer test. And we use the 2-key HSD function built into R. We simply insert the name of our Anova model, model.1, into our 2-key HSD, and it spits out at us this output. And as you can see, we have our group species, and we have our comparisons, versicolor setosa, virginica setosa, and virginica versicolor. The only thing you need to pay attention to is the p-adjusted value here. If the adjusted p-value is less than 0.05, then the difference is significant. In this case, yeah, all 3 are different from each other. You might be curious what a adjusted p-value is? Well, when you test a set of data more than once, you lose degrees of freedom. And your chance of making a type 1 error goes up. So a p-value from a post hoc test needs to be adjusted to take into account the multiple testing that you've done. There are many ways to adjust a p-value, but just know for this particular test, the adjustment is made based on the fact that we are making one analysis, and that's the Anova, and then we've made 3 more analyses, which are the individual comparisons between groups.